All right, so now we're going to get into one of the major physiological functions in the vascular system, and that is capillary exchange. So we're going to talk about the different mechanisms that occur in the tissues. We're going to talk about filtration, reabsorption, hydrostatic and osmotic pressures, and then net filtration pressure, and talk about some of the physics laws and chemistry laws that'll apply here. So remember, the capillary wall is literally just like one cell thick, which means there's a couple ways we can transport stuff. One is diffusion. We just transport from one side to the other through that single cell. Normal diffusion through carriers. Transcytosis, where we go through the endothelial membrane, and then bulk flow, we're going to filter and reabsorb across the endothelial membrane. Transcytosis, this is for larger substances, right, where we're using substances enclosed in a vesicle, and then we're using basically endo and exocytosis to bring things into the cell, transport them across, and then have them exit out. Again, this is for things that are really big that typically cannot just diffuse through a membrane. So antibodies, albumin, insulin, usually we're looking at things that are large and protein-based, things that are soluble in water. Diffusion is when we're talking about things like gases, oxygen and carbon dioxide, as well as other things that are much smaller, like glucose and amino acids and certain small hormones that can pass right through. These can pass through capillary walls, right? So oxygen, carbon dioxide, these are both, they're able to pass through. They don't have charges or anything, so we don't have to worry about that plasma membrane being in the way. The one difference is when we look at the blood-brain barrier, it's going to be a bit more strict. Now, bulk flow, this is so diffusion, we're looking at plasma interstitial fluid, where bulk flow is big stuff. We're talking moving lots of water. And this is what's going to help us with maintaining volumes of blood and the fluid between the cells. Again, I mentioned this in the past video where capillaries are leaky, meaning liquid is going to move from the bloodstream into the tissue space, and we're not necessarily going to reabsorb all of it. And so the movement of these things, right, through these leaks depends on hydrostatic and osmotic pressures. The fluid from the blood moving into the interstitial space is called filtration. And again, we do not have fluid building up massively because as it drops in the capillaries as we pass through, the osmotic pressures are going to switch, which is going to cause reabsorption into the capillaries. Again, it doesn't pull everything up, and that's where the lymphatic system kind of picks up the slack. So filtration, this is pressure driven of fluid and solutes from blood to interstitial fluid. So the blood pressure and the interstitial fluid osmotic pressure are going to promote filtration, and then reabsorption, right? This is the movement of fluid and solutes from the fluid back into the blood capillaries, right? And so if we look at kind of what's happening here, right? Here's the artery side, here's the venular side. As we're moving through, right, hydrostatic pressure is pushing out, right? Whereas as we move down here, the oncotic pressure, the pressure from the tissues is higher, and so things get pushed back in. Bulk flow depends on 4 pressures. Blood hydrostatic pressure. This is to push fluids out. On the arterial end, the blood hydrostatic pressure is higher. It's about 30 milligrams of mercury. On the venous end, it's dropped down to 10. And so this is why as we enter into the capillaries, things are going to be pushed out, right? Because the blood hydrostatic pressure is higher on the arterial end. The interstitial fluid osmotic pressure is the force of osmosis out of the capillary because of there's small proteins that have leaked into, that are basically pulling fluid, right? There's proteins that are drawing fluid out. That's about 8 milligrams of, or millimeters of mercury. The interstitial fluid hydrostatic pressure is the pressure of the fluid pressing on the outside of the wall. This is going to push fluid into the capillary, but we notice it's low, right? 0 to 3 millimeters of mercury. And then the blood colloid osmotic pressure, this is from the bloodstream's plasma proteins in the capillaries. That's going to pull fluid in, right? And that's 28 millimeters of mercury. And so it's the combination of these things, like as we talk through these, we'll see how these numbers will, or these different types of pressures will act on a single capillary to move things out of the capillary and then also move fluids back in. But the big thing, no matter what, is that if the outward forces are bigger than the inward forces, so forces pushing stuff out are bigger than the forces pushing things in, then things are going to flow out of the capillary. This is filtration. If the inward forces, things pushing fluids in, are greater than the forces pushing things out, then this is reabsorption. We're going to push things back into the capillaries. And so we have our basic kind of function, I guess we'll say, or equation of net filtration pressure, or NFP. Net filtration pressure is going to be our BHP plus our IFOP, which is all the outward pressures, right? These are things pushing things from the capillaries out to the tissues, minus the things that push inward, the BCOP, the IFHP. Those are both pushing things back in towards the blood vessels. If the number ends up positive, right, then it's pushing things out. If the number ends up negative, we're pushing things in. And what we notice is that on the arterial end of a capillary, the side of the capillary that's closer to the arteries, we tend to see a positive number, meaning things are pushed out. On the venous end of the capillary, we see that the numbers have gone more negative. And so because of that, things are generally pushed back into the capillaries. So here we go. So we see, making our way through, right? Here's our blood hydrostatic pressure, or BHP, it's 35. Our BCOP, our blood colloid osmotic pressure, is 26, right? 35 plus the 1 IFOP, right? So 35 plus the 1 is 36, 26, and we have nothing going on the other end, right? Our IFHP is not there, and so we see that 35 plus 1, 36, minus the 26, it's 10, right? It's a positive number. We're filtering out. Things are being pushed out more. As we make our way across, right, our blood hydrostatic pressure has gone down, right, from 35 to 16, so 16. The 1 is still there, so 16 plus 1, 17. 26 BCOP, and we don't see anything with the IFHP, right? So it's still 26. So now it's 17 minus 26, we have a negative number. More stuff is being pushed into the capillary in comparison. So normally, filtration absorption is about equal. We're what we call equilibrium. This is called Starling's law of the capillaries. And again, it's using that specific calculation, the NFP equals BHP plus IFOP, all minus BCOP plus IFHP. So this should normally, like as we look across the capillary, this should zero out. It should be about equilibrium. But if we see a disruption in that, this is where we can end up with something like a lymphedema or an edema, where we have more fluid accumulating in the tissues.